Hi, and welcome to the third part of our epigenetics section. In my previous videos, we have discussed what epigenetic actually studies and how the Mendelian randomization approach can work with epigenetic data, mostly DNA methylation, just because that's what we have available technologically. In this section, we will go through some examples of pretty well done Mendelian randomization studies that actually use DNA methylation data. And my hope is that it will provide you with a sample workflow for approaching your own research questions. This video will use data from two very recently published papers. The first one offers an analytical framework that integrates high-resolution genotype data with DNA methylation data to support causal inference. We will focus on how they established that specific CpG sites can mediate the relationship between the genome and downstream cardiovascular risk. In the second example, we will consider a different epigenetic marker called the epigenetic clock, which is a way to capture your biological aging using also DNA methylation data. And it turns out that the rate at which your body is aging is at least somewhat under genetic control, so we have a potential instrumental variable for that, for this very interesting construct of epigenetic age. I have provided both links on this slide, so feel free to click and follow along or look up more details. The main question of the first study was whether DNA methylation that was measured using the Illumina chip on a genome-wide scale was on the causal pathway between the genome itself and various cardiovascular traits. Now that might seem more or less familiar to you because that's the causal scenario that we discussed in the two-step method, and that's summarized here. That method can help us test mediation of exposure-disease relationships by epigenetic markers. It's really the same question. But there is somewhat of a difference in this example because the exposure is the genome. So the exposure and the SNP in this diagram, particularly in step 1, are the same thing because we're interested in the mediation of genetic effects themselves. So that actually makes our job a little bit easier. Let me show you how. Such a study, assuming that we see a significant association, would have several possible causal outcomes. In the simplest one, and that's the one shown here, the genetic variant has an effect on the phenotype, and that effect is mediated by DNA methylation. This is the hypothesis we're testing. That SNP in question is our MEQTL, and it serves as both the exposure and as the instrumental variable for DNA methylation so that we can get all those benefits of random assignment that we get with Mendelian randomization if our assumptions are satisfied. The second scenario is in the reverse direction, also very familiar to you by now. In this case, we'll have a bunch of instrumental variables, and those are no longer MEQTL instrumental variables. Those are instrumental variables for your outcome, so they're causing variation in the trait. In our case, it's a bunch of cardiovascular traits. And that variation in turn influences DNA methylation, just like we saw in the lipids example in the previous video. That could happen. And in this case, the relationship between the genome and the outcome is not mediated by epigenetic changes. Something else is going on. So that is also a plausible causal explanation. But we're not even done yet. The third possible causal explanation also says that DNA methylation is not a mediator of this relationship between the genome and the cardiovascular trait. Instead, here, we have two SNPs that are not independent. They're in linkage disequilibrium. So there's a correlation between them. And one is influencing DNA methylation, while the other is influencing the outcome. So you might see an association between the DNA methylation and the outcome, but it's because of this phenomenon. Finally, we have the fourth scenario. In that case, the same SNP causes changes in both the DNA methylation and the cardiovascular trait. That is not at all what we want because that's pleiotropy. It violates the third assumption of instrumental variable analysis and it makes our estimates of causal effects invalid. That is something that we worry about in epigenetic studies. Just how invalid can it render our estimates? I linked a paper in this slide that shows that it can skew estimates as much as 200%. And it also shows that you don't always have fancy statistical methods to correct for pleiotropy. But you might as well know about it, so it is also a scenario worth testing. So now we need to figure out of those 4 possibilities, which one is actually consistent with our evidence? Which one is our best guess of truth? What's really, really nice about this paper is that they came up with an algorithm to help you arrive at an educated decision about which scenario you're actually facing here. The first thing they did was choose a bunch of SNPs that they knew to be associated with DNA methylation. And there are many of them. We're talking about hundreds of thousands of them. In my next video, I will show you exactly which database they used to do that. So they tested all these SNPs for association with a number of cardiovascular traits, things like inflammation, lipids, BMI, and so on. They found some associations between these MEQTL and cardiovascular disease traits. That's step 1. Now, if they didn't find these associations, they just threw away those MEQTL SNPs. But if they did, remember that this association actually can't be treated as causal because the exposure, in this case the MEQTL, is a SNP, it's randomly assigned. And the authors found such evidence for 10 MEQTL SNPs that they then took into the next step. So let's say from the first step, we do know that methylation is causally affecting the phenotype, because they did, right here in that second box, they did a Mendelian randomization study and confirmed that it is in fact causal, that all the assumptions are satisfied and so on. So now we can ask our second question in the red box, and we can do that in another sample. The question we ask here is whether the phenotype also had an effect on methylation. That's scenario number 2. Here, we will use a different instrumental variable, and those could be SNPs in other data sets that had been shown to cause variation in those cardiovascular phenotypes. And here we have 2 possibilities. We can either have strong evidence that the phenotype is influencing methylation. That's what we saw in the lipids example. We've seen it before. And that would lead us to this reverse causation scenario. But that is not at all what happened in this paper. Granted, they were underpowered to test this particular effect, but they also did not see any causal effects from the cardiovascular phenotype to the epigenome. So for now, we can rule out that possibility. So we follow the no arrow, and here is where the authors talked about find mapping. Find mapping is not really the focus of our course, but essentially it is a technique that you can use with high-resolution genetic data to estimate the probability that your SNP is in fact the causal variant in that region. And you can do that for multiple causal variants. The authors used this technique to rule out the explanation that said they're actually separate SNPs. One's influencing methylation and one's influencing the phenotype, and they just happen to be in linkage disequilibrium. Remember, that was scenario number 3. So if they found discordant effects like that, they concluded that this kind of LD-mediated structure was true. But that's, again, not what they found. What they found was pretty good concordance for some of the traits, not so much for others, and for some traits they could not do this analysis because they didn't have enough evidence. So, assuming that we do in fact see concordance here, we go into the final step of ruling out pleiotropic effects, scenario number 4. If we can do that, great, because that means we have evidence that epigenetic variants in fact mediate the relationship between the genome and cardiovascular traits. If not, Well, at least we know that we're dealing with pleiotropic effects, and either way, you're generating interesting evidence. The authors proposed evaluating this by using multiple instruments, which makes sense, right? If we use multiple instruments, we far reduce the likelihood of having the scenario on the very left for every single one. But unfortunately, in the case of this paper, and in the case of epigenetic studies in general, we usually only have single instruments valid for each methylation locus, so we cannot really test for pleiotropy. You need multiple instruments. And that is unfortunately very common in epigenetics. Why? Because usually you only have one independent MEQTL per CPG site. If there is another SNP associated with your CPG site, it's probably in linkage disequilibrium with the first one. So they're not independent. Usually there's only one independent instrumental variable, and that makes it really difficult to reliably discern between true mediation and horizontal pleiotropy, which is what we're seeing in scenario 4. Nevertheless, this paper really offers a thoughtful step-by-step approach to disentangling causality in the epigenetic context, and I hope you find it useful in your own work. The second example that I wanted to discuss today also uses DNA methylation data, but in a very creative way. Turns out that there is a way to take a handful of CPG sites from the Illumina array and check if your biological aging process is faster or slower than your calendar age would suggest. So for example, you know you're in your late 30s, but depending on what your exposure history has been like, the methylation status of this handful of CPGs can tell you what your real biological age is. We know that offspring of people who live to be 100 tend to age slower. And we also know that people exposed to certain bad things for you tend to age faster. And this tool is strikingly accurate. It also shows that those who are aging faster are also, unsurprisingly, at much higher risk for certain diseases. If you want to play with this calculator, it is freely available, and here's the link. There are two epigenetic clocks. One is called the intrinsic clock, and the other one is the extrinsic clock, and they use different sets of CPGs. The extrinsic one is a better marker of lifestyle, and the intrinsic one captures more the fundamental aging process intrinsic to the cell. And you can do that in different tissues. Those two clocks, intrinsic and extrinsic, are really different constructs, and they're only moderately correlated in most tissues in humans. Using both intrinsic and extrinsic clocks, we can think of epigenetic age acceleration, which is usually a bad thing. That means you're aging faster than you're supposed to. And that's associated with a variety of chronic diseases and mortality, as you can see here. Or deceleration, that's a good thing. That means your body's young for its age, or at least is aging slower. And that's associated with the usual suspects like healthy diet and exercise. Here are some more phenotypes and exposures that were observationally shown to associate with increases in the biological rate of aging. Turns out that things like eating fish and vegetables and having a healthy lifestyle that's stress-free, wouldn't that be nice, make you age slower. And also, both aging clocks are related to a bunch of cardiovascular and metabolic risk phenotypes. But we know with any epigenetic measure, those are just observational associations, and we cannot be so sure that they're causal. Maybe it's not the methylation that's causing all those outcomes. Maybe it's the other way around. And we know, again, we've seen that happen. So, of course, we go to Mendelian randomization. Instead of finding instruments for each individual CPG that's part of this clock, the authors of this paper just ran a GWAS using the two epigenetic age acceleration measures as the outcomes, and they had a really big sample with a wide range of age. and they found 5 potential regions of interest for the intrinsic one and 3 for the extrinsic one. As a side note, the rate at which you age epigenetically appears to be at least moderately heritable, as you can see here, and that can tell you some, but not all things about your own life expectancy, if you know the life expectancy of your biological parents, or rather the actual life duration of your biological parents. So they found those loci, those SNPs that are associated with aging, and then they did something interesting. For each of those loci, they used Mendelian randomization to check whether gene expression, now it's not methylation anymore, but it's the actual expression of the gene in the region, measured things like transcriptomic arrays or RNA-seq, was causally linked to the epigenetic aging process. And they found that to be the case for 6 genes. Now, not all of you will have expression data. Do not despair if you don't. And remember, with Mendelian randomization, you can do things in separate samples. And there's some public gene expression databases available should you be interested in pursuing this strategy. One of those 6 genes that this particular study causally implicated in aging was called TERT, and that stands for telomerase or reverse transcriptase gene. It governs telomere length, and that's yet another marker of biological aging. But what's really interesting is that there is no evidence of a causal relationship between the telomere length and DNA methylation age. The authors of this paper ran the Egger regression and found some pleiotropic effects for the TERT gene. What's even more interesting and kind of paradoxical is that the effects were in opposite directions. What helps telomere length apparently hurts you in the epigenetic age milieu. That tells you some very curious things about biology, and the authors followed up on Mendelian randomization by looking at it in experimental models. In Mendelian randomization studies, you would want to be very careful with this evidence because clearly you have pleiotropy. The fact that these 2 measures of aging, telomere length and the epigenetic clock, are so unrelated, statistically speaking, but they have the same genetic causal root, that means that the environment matters a lot more for how quickly you age compared to what your genome says. For some people, that's great news, and for others, not so much. The final note about this particular study is that it also found, through another round of Mendelian randomization, that early menopause and early menarche cause quicker epigenetic aging, and that's consistent with the prevailing hypothesis in the field. So, today, we went through 2 examples of pretty cutting-edge epigenetic studies that use Mendelian randomization. I hope that shows you the diversity of approaches that's possible in our field, and I hope it inspires you to do some work in epigenetics of your own. And of course, I hope that you will use Mendelian randomization creatively and appropriately. In the next video, I will show you some web-based tools that can help you do it. Thank you, and have a great day.